Hello, everyone. I'm sorry I can't be there in person today, but thank you for the organizers for being flexible and letting me present virtually. I'm Alex Maris. I'm a postdoc at University of Chicago in Matt Terrell's lab. And this talk is about physical property scaling relationships for polyelectrolyte complex micelles. So the situation that I'm particularly interested in is the case where we have a block copolymer, a block polyelectrolyte, and a homopolymer polyelectrolyte. And these assemble into a micelle or a nanoparticle, that has a complex core of neutralized polymers, and then a corona surrounding that core that's consisting mainly of the neutral, what I'm calling here is the A block. And I'm interested in this system mainly because it's shown to be effective for drug delivery. So I'm just showing a couple snippets here from our group and collaborators and the Karioka group in Tokyo that are using this system to deliver a therapeutic RNA. In both cases, they're using PEG polylysine as the block polymer, and then some sort of a microRNA inhibitor or an antisense oligo. And these have shown to be effective in cells and in mice. And actually, just a recent update from our group is showing that using PEG polylysine and a targeting peptide can sequester this microRNA inhibitor into a micelle. And then actually deliver this and target atherosclerotic plaque and show a significant decrease in the size of this plaque after it's delivering the RNA inhibitor. But this isn't really the point of my talk today, but this is just to show that these micelles are effective for therapeutics, but we don't really know how we can control the size and the physical aspects of these micelles, even though we know ultimately that it's important for the therapeutic applications. So this is something that I'm interested in largely throughout my postdoc, is how we can use different aspects of the polymers that are going into these micelles to control the nanoparticle structure. And so for this work, I'm using PEG polylysine here in every case for this ABE block copolymer. And then I'm using polyglutamic acid or single-stranded DNA for different cases as the anion. So we're mixing these together in solution and actually putting these in a very high salt condition, and then slowly removing that salt through dialysis. This assembly process sort of anneals these to get them to a low equilibrium state. And then we're characterizing these with a few different methods. Electron microscopy that I'm showing here on the left. This is all for a couple different cases that I picked from this study. Electron microscopy that's showing some core information. Small angle X-ray scattering. This is most of what I'll talk about today. This is done at Argonne. This is also the core size information across a couple orders of magnitude of size, which is really powerful. And then dynamic light scattering, which is giving us the hydrodynamic radius, or this larger like whole size of the micelle. And so we've spent a lot of time over the past few years, a lot of different people developing these assembly methods and characterization methods and learning how to properly fit this data to the point where we feel very confident in the accuracy and the reliability and repeatability of these results. And so this is what really enables us to do this sort of scaling relationship study. And so let me show you some of the results here. This is the first example. This is core radius, so this comes from small angle X-ray scattering. And this is, I'm basically plotting the size here versus the length of each block. So this might be kind of cut off here, but this is the A neutral block, the B block, the block polymer, and then C is the homopolymer length. And so, this is one data set for one homopolymer, but we have 5 of these sets, except for this case, this is 2 data sets. But basically we're fitting this data to a power law. This seems to fit well, and this is consistent with theory for polyelectrolyte complex micelles and also other polymeric nanoparticles. And then what I'm showing here on the right is a scatter plot of all the exponents of these power law fits. So for instance, one of these blocks here is this exponent from this data. Another block is this exponent, and the others are the rest of the data that I'm not showing here. And these are, fairly consistent. And so if I'm averaging all these exponents, that's how I'm coming up with a scaling law for this system. So what we see here is for the neutral block, as we increase the A block, or the PEG in this case, the core size is getting a little bit smaller, slightly smaller. As we increase the B block, or the charge block of the block copolymer, the size is increasing much more drastically. And then as we change the C block, or the length of the homopolymer, which is either polyglutamic acid or DNA, in every case, it's pretty much nothing is happening. This is an independent relationship. So next, I'm looking at the hydrodynamic radius. So this now is coming from DLS. And again, this is now the size of the whole micelle, including the corona. And I do the same strategy here with our DLS data. And what we find is, and also if we have the hydrodynamic radius and we have the core radius, we can compute the corona thickness for each case. And so I'm not showing it here, but I calculated the thickness for every data point, and then went through this process again to get scaling laws for both the hydrodynamic radius and also the corona thickness, with some propagated error for the thickness. But what we see is that as we increase the neutral block, we have a slight increase in the hydrodynamic radius, a more drastic increase in the corona thickness, as you expect. This is the neutral A block. Similarly, we have an increasing hydrodynamic radius with the B block, which I think mainly comes from changing the core size, like I showed before. And again, really no effect from the length of the homopolymer. And then the third physical parameter that I'm looking at here is aggregation number. And so these data also come from small angle X-ray scattering, which is where I got the core size from. But this is a little more complicated to get from that data. The length of the B block and the length of the C block that form the core are not the same in most cases. So if I'm trying to calculate aggregation number, you can extrapolate the intensity to Q equals 0 and end up calculating something. But what I'm actually calculating here is the number of ion pairs in the core of the micelle. And so you can kind of think of this as like a monomer aggregation number, maybe. But if I'm assuming that the core of the micelle is completely neutralized, and so the number of cationic monomers is the same as the number of anionic monomers, which is the same as the number of ion pairs, and I know the length of each polymer, then I can calculate the aggregation number for each polymer. And that's just the simple relation here. So what I'm showing up here is the scaling for the number of ion pairs in the core. And then what I'm showing in these two right here is the aggregation number for each polymer. And so what we see is that increasing the A block we see a slight decrease in aggregation number. As we increase the B block, we see a very drastic increase in aggregation number. And like everything else I showed so far, there's just no effect from changing the homopolymer length, the C length. And so to summarize here, this is all the scaling laws that I showed today. And this is a lot of numbers here, but the main message is that the B block, or the charge block in the block copolymer, is really driving most of the change here. It's controlling the size of the nanoparticles and the aggregation number. We're seeing a slight effect of both of these from the A block, and then across the board on all these physical parameters that we looked at, we're seeing no effect from the C length. But this is actually really nice, because that C block in most cases is our therapeutic, right? And so if we're trying to deliver this, we want to control the size, and we don't want to change our therapeutic, well, the fact that it doesn't affect anything here is really convenient, because we can just throw it in and it should be controlled by the polymers. So that's kind of nice. And so now going to literature, published work from our group and from other groups, looking at anything I could find where somebody was using this AB plus C polyelectrolyte system and varying the block length. And so I'm plotting all of that here, and I'm normalizing all of this using one of our scaling laws to compare to the other scaling law. So like, for instance, here, I'm looking at the A block, so I'm normalizing it for the B block, and everything collapses pretty nicely and matches with our scaling laws that I'm overlaying here in black. And so this includes synthetic polymers and biological polymers and different neutral blocks, in the case where the B block is anionic and the C block is cationic. It's really nice to see all these different situations still, consistent with these scaling laws. And so the last thing I want to compare it to here is the theory. And so this is theory specifically for polyelectrolyte complex micelles, and they look at the two extreme cases where you have this crew cut situation where the core is much larger than the thickness of the corona, and the other extreme is the star-like, which is kind of the opposite. And experimentally, our micelles are well between these limits, and our scaling laws are also mainly between or close to those limits, which is really nice to see that agreement with theory. And so to wrap up, these are the first experimental scaling laws for this system, and it's really nice to see them show some agreement with theory and with the previously published data. This is an interesting study for polymer physics, but it's also, I think, really important to have this understanding for delivery, for the therapeutic applications. We know that the size and the stability and all this structure is important for biodistribution and ultimately uptake and release. And so if we can predict and control that structure, this is going to be really important in the future for those applications. So this is something that I'm interested in for this work and for moving forward, both for polyelectrolytes and other polymeric nanoparticles, where we're also going to need this sort of understanding. So with that, I just want to thank everybody for listening, hopefully. And I want to thank my lab and the Trinity undergrad student and some other people that helped in direct and indirect ways, and then Artem especially for many conversations about his theory. I can't take questions, but this is published now if you want to read more, or please don't hesitate to email me about anything. So thank you all.